Welcome back to Upregulate. Today we discuss gene therapies. How do they work, how expensive are they, and are they safe? First we tackle, how do gene therapies work? Genetic science has taken over a century to develop into current gene therapy advances. The Austrian monk Gregor Johann Mendel was a pioneer in genetics as he studied the patterns of inheritance by analysing individual units, now known as genes. James Watson and Francis Crick proposed the double helix structure of DNA in 1953, which is known as the dsDNA model. This model provided a physical explanation of how genetic information is stored and passed on, revolutionising the field of genetics and molecular biology. In 1970, scientists discovered enzymes that could separate genes at specific locations on the DNA strand, leading to new opportunities in gene therapies. And recently, gene therapies have emerged as a cutting-edge approach in molecular medicine, with great potential to treat genetic disorders. Today, gene therapies have already been used to clinically correct the genetic alteration in some inherited disorders, such as haemophilia, cystic fibrosis, thalassemia, RPE65 gene-related retinal diseases, and is also used to tackle acquired diseases such as AIDS, Parkinson's, cancer, and Alzheimer's disease. Many diseases result from genetic mutations and cannot be treated with traditional medications or require long-term treatment of the symptoms. However, new gene therapy treatment methods allow us to target the underlying genetic causes. Gene therapies are a powerful technique that aim to correct genetic mutations in these disorders by manipulating the genetic changes at the core of the disease. Gene therapies encompass several approaches for treating genetic disorders, including gene silencing using RNA interference techniques such as siRNA, shRNA, and miRNA. Gene replacement is also another method through the direct administration of plasmids or viral vectors, and gene editing using specific nucleases like ZFNs, TALENs, and CRISPR-Cas. Put simply, there are three methods: silencing, which interferes with the expression of a certain gene, therefore silencing its expression; replacement, which replaces a faulty gene or adds a new gene; editing, a one-time therapy that directly edits pieces of DNA within the cell. Honestly, I can probably go on forever about how gene therapies work, and I can and I probably will make a video covering the no-holds-barred ins and outs of gene therapies, current delivery methods, how each type works, etc, etc. In the meantime, if you want more information on this subject, I highly recommend you read this paper titled Gene Therapy, Comprehensive Overview and Therapeutic Applications. This paper really tells it all. Next, are gene therapies expensive? Zolgensma from Novartis is a drug that carries a list price of $2.125 million per treatment. The price tag of some gene therapies can reach into the millions of dollars per dose. However, these costs have been justified according to their expensive production costs. According to this article and Eric David, the CEO of Therapeutics, the cost of goods/manufacturing alone for gene therapy have been estimated to be between $500,000 and $1 million, and that's not including the cost for research and development, the costs to run clinical trials, or the costs to build the commercial infrastructure necessary to provide access to patients. For now, many of these therapies are administered as a one-time only treatment, and they're often administered to very small patient populations, as many of the conditions treated are extremely rare, sometimes affecting as little as 100 people worldwide. With the low demand and high costs of delivering these treatments, the associated price may be considered justified to relevant pharmaceutical providers. Are gene therapies safe? Like most other treatments, gene therapies undergo strict clinical trial testing that take decades to complete. This long trial period allows scientists to understand both the short-term and long-term effects of the treatment, and develops a picture of how safe these products actually are. The ethical considerations surrounding human applications of gene editing have been broadly divided into two categories: gene therapy, which aims to correct faulty genes, and genetic enhancement, which aims to improve normal genes. One area of controversy is that the type of cells being edited whether it be somatic or germline cells. Editing somatic cells, which are non-reproductive cells, is generally considered ethical, as the impacts will not be passed down to future generations. However, editing germline cells, which are reproductive cells, is generally considered unethical and immoral, as the changes will pass down to future generations. Article 24 of the Universal Declaration on the Human Genome and Human Rights, adopted by the General Conference of the United Nations Educational, Scientific and Cultural Organization, states that germline modifications could be contrary to human dignity. Gene therapies have the potential to eradicate heritable genetic diseases. However, social, ethical and religious concerns have kept research on human germline gene editing strictly regulated, while clinical applications are currently prohibited in more than 50 countries around the world. In this paper, referring specifically to CRISPR-Cas9 ethics, gene editing technologies such as CRISPR-Cas9 carry two primary ethical concerns for the future of gene therapy method. One is that there could be an unforeseeable or unidentified risk in germline gene editing, essentially causing potential off-target editing of non-target genes. And two, the possibility that some ill-intentioned organizations or individuals could use gene editing technologies towards eugenics, i.e. editing of specific genes in individuals to yield specific traits. However, issues in gene therapy targeting can be honed and tested in clinical trials, and the theory of eugenics applications is considered far-fetched at this point. Regulatory frameworks will be established to correctly monitor these technologies worldwide, and agreements such as the Universal Declaration on the Human Genome and Human Rights help to stop exploitation of gene therapy methods. Although gene therapies have promise and some are now commercially available, a large quantity of gene therapy methods are in early development stages. These treatments have passed through these rigorous and lengthy clinical trial periods and have been deemed safe for use. Thank you for watching again today. I hope you've enjoyed the video. If you want to see a bigger video on gene therapy methods, I can work towards making that. I think it's something that I'd like to invest more time into and learn more about, including each individual method of gene therapies. So I can speak about each of them. If that is something that is interesting, please like and subscribe if you like the video, and I'll see you in the next one.